Cell and its environment. This involved the interaction between a cell and its environment. It is discussed as two major subheadings: active transport, passive transport. Active transport. This involved the movement of molecule or cellular energy in form of ATP against concentration gradient. Example of processes that involve active transport include phagocytosis of bacteria by macrophages, polarization of neuron. Passive transport. This involved the movement of molecule or ions into a cell and its environment without utilization of cellular energy. Passive transport is divided into diffusion, osmosis. Diffusion. This is the movement of molecule, solid, liquid, gas from a region of higher concentration to a region of lower concentration until they are evenly distributed to reach dynamic equilibrium. Example: absorption of mineral by plant roots, movement of hormone and enzymes through the bloodstream, absorption of nutrient by the villi of the small intestine. Factors that affect the rate of diffusion: temperature, size of molecules, concentration gradient, state of matter. Temperature. This is the function of average kinetic energy of the molecule. An increase in temperature increases the average kinetic energy, which also increases the rate of diffusion. A decrease in temperature decreases the rate of diffusion. Size of molecules. The smaller the size of the molecule, the higher the rate of diffusion. Concentration gradient. Concentration is the amount of solute present in a substance. Diffusion increase with a high concentration gradient and decrease with a low concentration gradient. State of matter. Diffusion is faster in gases, fast in liquid, and slower in solid. Osmosis. This is the movement of liquid molecule from a region of lower concentration to a region of higher concentration through a semi-permeable membrane, in order to establish ionic equilibrium. Example: movement of water away from the red blood cell. Terms used in osmosis. Hypertonic solution. This is a solution whose concentration is higher than that of cell content. Example: salt solution, sugar solution. When a cell is placed in a hypertonic solution, it tends to lose water to the surrounding, which appear shrunken. Hypotonic solution. This is a solution whose concentration is lower than that of its leaving cell content. Example: distilled water. When a cell is placed in a hypotonic solution, it tends to absorb more water from the solution and become fully stretched or fully expanded. If it's an animal cell, it will eventually burst, but if it is a plant cell, it becomes fully expanded and does not burst due to the presence of a cell wall. Apply a pressure on the cell content called turgor, or wall pressure, while the cell content apply pressure on the cell wall called osmotic pressure. Isotonic solution. This is a solution whose concentration and that of its leaving cell are the same. Plasmolysis. This is an outward movement of water from a plant cell when it's placed in a hypertonic solution. Note: in plasmolysis, water is gradually loose from a cell, while flaccidity water is rapidly loose from a cell. Flaccidity. This is a shrinkage of a plant cell as a result of loss of water from a hypertonic solution. Turgidity. This occurs when a plant cell absorbs water from a hypotonic solution and become fully stretched or expanded in order to carry out various metabolic activities. Hemolysis and crenation. Hemolysis occurs when red blood cell, erythrocyte, absorb more water from a hypotonic solution and eventually burst. Crenation is a shrinkage of a red blood cell after it has lost water to a hypertonic solution.